Hello, everybody. We are back for part two of the Pons. This is the slide where we stopped, so we're going to begin here and go forward. And as you may recall from the outline, we covered the caudal pons last session, and today we're going to cover the rostral pons, including cranial nerves 7, 6, and 5, as well as all of the structures in the rostral pons. So let's get going. Starting with the facial nerve. So the facial motor nucleus provides special visceral efferents to the facial mimetic muscles, and that it's called special rather than somatic because it's off the branchial arch 2. This is also famous for its loop around cranial nerve 6, and we're going to look at that. In addition to the motor component, there's also a general visceral efferent from the superior salivatory ganglion and the lacrimal nuclei that are going to contribute to the preganglionic parasympathetic axons through nervus intermedius and to the pterygopalatine ganglion for lacrimal secretion, and then through the chorda tympani to the submandibular and sublingual glands. And we'll look at those. And the location of the superior salivatory nucleus, which we really haven't looked at, but we will, it's just a bit rostral to the dorsal motor nucleus of the vagus. You also have these special visceral afferents from the tongue, which are going to the rostral nucleus solitarius, therefore, of course, for taste. And that's going to travel via the chorda tympani. You have a minor general somatic afferent from the ear to the spinal trigeminal nucleus that carries the somatic sensation from the anterior wall of the external auditory canal. And then you also have the, I just want to point out, not part of the facial nerve, but I want to point out that the posterior third of the tongue sensation for taste, as well as for general somatic sensation, comes in through cranial nerve 9. Also that the general somatic sensation from the anterior third of the tongue comes in through the mandibular division of the trigeminal nerve. So we're going to look at all that, and most of this is very familiar to us as we take care of patients and examine patients who come in with a Bell's palsy. This is in the medulla, but I just put this up to remind you of this columnar organization of the general somatic efferents, the general visceral efferents. Those we're going to be dealing with. Motor nucleus of 7, motor nucleus of 5 also will be in this column. The general visceral afferents and special visceral afferents, and then out to the somatic and general somatic, special and general somatic afferents. So here we are. I use both of these diagrams for you. I like the color coding of this one, but I frankly like that this one separates the motor root of 7 from the nervus intermedius. Otherwise, this one doesn't really deal with the nervus intermedius. And then this is particularly important when you want to ask yourself, what's the relationship of nervus intermedius to the chorda tympani? And it has to do with their relationship to the geniculate ganglion, okay? And so chorda tympani is distal to that, and the nervus intermedius is proximal to that. This is also a good diagram here in the lower left, because it also shows you the salivatory nucleus, which is not labeled, but it's sitting right here. If we then come back up to the easier to see color coding, we see the motor nucleus of 7 looping around cranial nerve 6, and then heading out the nervus intermedius, of course, not participating in the ganglion, but continuing on into the motor root of 7. And then you can also see here, if you sort of trace backwards to the yellow, which is tough to see, from the salivatory nucleus, heading out to the pterygopalatine ganglion and down to the submandibular ganglion. And then finally, you have the, demonstrated here in the blue, as the special afferents for taste, making their way up. Notice that the cell body is here in the trigeminal ganglion. All right. So let's look at this in some slides. Cartoon. Here's the motor nucleus of 7, or its location, middle cerebellar peduncle, just for orientation, right? Here's the superior salivatory nucleus and also the lacrimal nucleus right next to it. This is the facial colliculus, which we see in the floor of the fourth ventricle. And this will be therefore the abducens, which we'll come to. And then this is nice because it shows you on this side how the nerve exits. This will be from the superior salivatory out the nervus intermedius, heading out to the ganglion. And here is the motor root coming out to the muscles of facial expression. And, I think it is a good idea, and I'm not going to hang out here or ask anybody to do this right now, but it's a good exercise, even after this talk, during the hour that you spend studying it, to think about why the question is not just what the afferents are, but it's to get you to think about what the afferents need to be in order for the facial nucleus to do everything that it does, and all the things that we just talked about. There's motor expression, there's other things that you are aware of. There's taste, of course, there's salivation and lacrimation, but there are also other things that it does, whether you're sneezing or blinking, right? It participates in the blink reflex. And it also is a protective reflex when there are extremely loud sounds. All of those things, therefore, have to have afferents to the facial nucleus. And so, of course, number one, you get direct cortical bulbar fibers for our facial expression, literally for our voluntary facial expression. To smile, to frown, to flirt, to show surprise, and everything that we do to convey nonverbal information has to be done using the muscles of the face. But then you have indirect cortical bulbar fibers, which go through the reticular formation. If you sneeze, if you take a bite of something, you don't want to bite your lip, right? That's important that you not bite your lip. But you don't want to think about that. And I'll go you one further. If you think about it, you will bite your lip, okay? One of the worst things I ever did in all of medical school was I was thinking, I was eating, and I was focusing on how my tongue works as it moved the food around my mouth. And I bit my tongue so hard that it didn't heal for about a week, okay? It was a mess, is all I can say. That's something you don't want to think about. And then you have some cross-bulbar fibers that are going to the upper half of the face, will be also for facial expression, and some involuntary facial expression as well. Then you will have secondary fibers coming from the spinal trigeminal nucleus to mediate the corneal reflex, okay? And secondary and tertiary because of the need to control the stapedius reflex, all right? And so it has to know that that's coming, right? In order to exert control over the stapedius reflex. And if you think about it, you also have facial expression when that loud sound is coming. All right, that's all I want to talk about with the 7. Move on to the 6. We've got a lot of ground to cover today. The 6th, the abducens nerve, is a column of neurons right near the midline, because these are true somatic motor, near the floor of the fourth ventricle. And we already talked about the facial colliculus. There are 2 populations of neurons within the abducens. There are the typical motor neurons that are going to innervate the lateral rectus. But then there are also these other group that are actually part of the abducens nucleus. These are internuclear neurons with axons that cross the to the contralateral MLF and ascend to cranial nerve 3. So these are going to be important for lateral gaze. They will be important for the oculovestibular reflex, doll's eyes, all right? And it's about 25% of the abducens neurons that work their way up to the contralateral cranial nerve 3 to innervate the neurons that will control the medial rectus. Remember, the cortico-bulbar tract has no direct input into the oculomotor nuclei. The abducens nucleus received afferents from the indirect cortico-bulbar fibers through the reticular formation that I just mentioned. Because they participate in this reflex for doll's eyes, they're going to have a medial vestibular nucleus component. All right, and they're also going to have the paramedian pontine reticular formation in addition, not the same, in addition to those 25% of neurons of the abducens nucleus, which will participate in doll's eyes. So a lot of doll's eyes innervation there. All right, let's look at it a little bit. Here is the root fibers of 6 coming out. Here's the nucleus of 6, right? Here's the root of 7 coming out. All right, here's the loop of 7. All right, so all of that coming in. We're gonna cover all the different pieces as we get to them. I don't want to do that right now. This is a good diagram. I like this diagram because it really focuses us on the MLF and look at how it enumerates all the various components. You have from the superior vestibular nucleus, you have a component ipsilaterally going to the oculomotor. You also have those fibers crossing into the MLF and ascending that are coming from the contralateral abducens. You also have a group that come from the ipsilateral, actually going to the contralateral medial vestibular nucleus, ascending, all of that ascending in the MLF. So I thought this was a very good summary diagram. And then there are, of course, the MLF descending fibers to the neck muscles, and we've previously discussed that. Well, I think this is a good diagram to keep things simple. All right, 7, 6. Now 5 is a lot more complicated. As complicated as 7 is, 5 is much more complicated. And 5 has the general somatic afferents from the face for touch, pain, temperature, from the face and forehead, from the mucous membranes of the nose and mouth all the way to the teeth, and also from the dura. So 5 is bringing in a lot of information, of sensory information for the head. Basically, almost everything in the head is coming in through the trigeminal nerve. It has the special visceral efferents from branchial arch 1 for mastication and related muscles of phonation and the oropharynx. You then have the trigeminal ganglion as an important part of this, Jussarian ganglion, semilunar ganglion, that sits on the surface of the petrous bone and then enters the tegmentum of the pons and ascends to the principal sensory nuclei or descends to the spinal trigeminal nucleus. We have previously looked at this in some detail, and so we're going to focus more on the ascending component today. This is a good diagram. Do me a favor, Varsha, could you turn off the light switch, please? So here is the Jussarian ganglion. All right, thank you. And so this is the trigeminal nerve. It's huge. It's massive compared to, here's 6 coming out over here. And you can see this is big. Okay. This is a great big nerve. You may remember that from the beginning, last week's talk, when we looked at the surface anatomy of the pons, how huge the trigeminal nerve is coming in. Now, just for some relationship, we're going to come back to the nuclei in a bit, but just to see some of the relationship, here's the 4th ventricle, right? Here's the superior cerebellar peduncle. We're going to look at those in a couple of minutes. Here's where the middle cerebellar peduncle here on the side with the embedded deep cerebellar nuclei, okay? Here's the juxtarestiform body. You may remember that was a reciprocal pathway between the vestibular cerebellum and the vestibular nucleus. All right, a little bit of the inferior cerebellar peduncle. Okay, and you can see where the middle cerebellar peduncle begins, and the 5th nerve is really running through that. The sensory nuclei of cranial nerve 5, there are 3 of them. We have dealt again with some detail on the spinal trigeminal, so I'm not going to deal with that anymore. We're going to talk about the principal sensory nucleus. Now, the principal sensory nucleus receives impulses for touch and pressure. Just the orientation of it is such that the ophthalmic division comes through the ventral component, or goes to the ventral portion of the principal sensory nucleus, the mandibular, so division the third division of the fifth nerve, will go to all the way to the dorsal portion of the principal sensory nucleus. But this is a peculiar nucleus. All of this, in many ways, is peculiar. The spinal trigeminal, because it goes down into the spine. But that was dealing, as you remember, with pain and temperature. Here you have these very large receptive fields with little adaptation. That's very strange for for light touch. That wouldn't make sense for light touch, okay? Which generally, we don't want that as I'm sitting down. I don't want all of this information coming from the skin that I'm sitting on right now, okay? I run or forget about this. And yet, there's no adaptation for this pressure and deep touch. And the way that I think about it, I always go back to phylogeny. And you have to remember that the trigeminal nucleus, the sensory portion, and in a certain sense, the motor portion, is a key element of survival in the more primitive phylogenetic forms. Because think of a mud puppy, you can't see, you're in the mud. So you're not seeing a whole lot. You're probably smelling things, so that's helping to guide you. But ultimately, you're going to have to put your face in what you're looking for. And you better not get bitten, and you better not get eaten, and you better not stick your head into a crevice where you can't get it out. That's why I think you have these non-adapting pressure sensations. You don't want to, and the vibresse that Puna talked about on the cat. You don't want to put your head where you can't get it out, okay? So these are important things, and they continue into our nervous, into the mammalian and human nervous system. In addition, you have the mesencephalic nucleus, which has driven me crazy all these years, because it is the only place in the central nervous system where you have cell bodies of primary somatosensory information. That is forbidden. You're always out in the dorsal root ganglion or in the Jusserian ganglion. There is no other instance. The only other instance where you have the cell body of this primary sensory neuron doesn't really apply. That's the olfactory bulb, which has nothing to do with any of it. So this is the only time that you have the cell body of the first order neuron sitting in the central nervous system. This is unique. And I have looked for decades for an explanation for this. And again, when I was preparing this lecture, I went back to the literature and thought, is there anything new? And the answer is, there is. And now, not an answer, but a clue. And in a paper that I was reading, trying to get to this, the author pointed out that actually there are two features that are unique of these neurons. One is what I'm talking about, its location. But the other, which I was never aware of or never thought about, was that what makes these primary sensory cell bodies unique is that they are the only cell bodies which receive synapses onto the cell body. The neurons of the cell bodies in the dorsal root ganglion do not have that, or in the Jusserian ganglion, but these do. And this is unique, okay? And so now, and I'm going to emphasize this as we go along, that these take on, because what are they doing? They're bringing in afferents from the spindles of the jaw closing muscles of the masseter temporalis and pterygoid. And so you have the jaw jerk, okay? A reflex that involves these. And this is sort of where I'm going. So let's back up. Let's look at the principal sensory nucleus of 5 is up here. Here's again the middle cerebellar peduncle. Here's the principal sensory nucleus, okay? And the mesencephalic nucleus we're going to see, but it's, you guess where? It's in the mesencephalon, so we're going to have to stretch to see it. Now, the motor nuclei, so let's continue and look at the motor nucleus of 5. These are typical anterior horn type cells, multipolar motor cells located near the principal sensory nucleus and near the mesencephalic nucleus. They lie medial to the sensory root. They receive collaterals from the mesencephalic root to create a 2-neuron reflex arc, okay? and they also receive corticobulbar directly. So just like before, when you want to talk, you're going to need direct control of the corticobulbar tract. When you're going to sneeze, bite, things of that sort, you're going to use your reticular formation or other local cranial nerve. And I just put this up here to show you the relevant muscles that we're talking about, and also to show you the periodontal ligament, which receives incoming sensory information into the trigeminal. And here's some more muscles, and I point out here, tensor tympani, the stapedius is cranial nerve 7. The rest of these are 5. And this is a great slide for showing us. So here we are, we're back to the, this is the same slide, but only a portion of it. Here's a sensory root of 5 coming in, this enormous root split up and then coming back. Principal sensory nucleus sits here, all right? Here, medial to that is the motor nucleus and the motor root coming out, all right? And here is the mesencephalic tract of the nucleus that will be going up. So this will be heading up to the mesencephalic nucleus. All right. This is good. This is several times now we're going to look at these crescent violet stains because, of course, here we are in the pontine nuclei. But here's the principal sensory nucleus of 5. This is in itself, this is a big nucleus. This is all by itself. Forget the spinal trigeminal, forget the mesencephalic. The principal sensory is a large nucleus. Here are the motor nuclei. At higher mag, they would look like anterior horn cells. And here's the tip of the mesencephalic nucleus. This will go up from here. So this is the caudal tip of the mesencephalic nucleus. Okay. A nice diagram, first of all, showing you again the size of trigeminal. Here we are. This is showing you the spinal trigeminal tract. This is these are the divisions of the primary. This would be of the spinal trigeminal tract, showing you how the ophthalmic, maxillary, and mandibular are aligned. Okay, ophthalmic being the most ventral. And here they come in, and here they are giving off their, some of these going down to the spinal trigeminal, and some of them going up to the principal sensory. They don't show the fibers traveling to the mesencephalic nucleus, but this is a good diagram to show you the location of the mesencephalic nucleus on that last crescent violet. We just caught the tip of those there. This is, again, another good diagram showing you, first of all, the cell bodies up in the mesencephalic nucleus. They're purposely drawn to look like the dorsal root ganglion neuron. Here's the motor nuclei drawn to look like a typical anterior horn cell. Here's the motor nucleus of 7 drawn the same way. Here's the hypoglossal drawn the same way. So you start to see a sense of integration. You also see how the spinal trigeminal nucleus is sitting in a location that will blend with Lissauer's tract and other portions of incoming pain information. And you also can see how this columnar organization works as you go up from the principal sensory nucleus sitting above the spinal trigeminal nucleus. Okay. There are the secondary trigeminal, in other words, from the nucleus. So we've been looking at the afferents to the sensory nuclei, but where do they go? What do they do? Well, mostly, of course, they're going to be heading to the thalamus, right? Because all of the sensory information, the somatosensory information, is going to have to get to the thalamus. Now, I've underlined the VPM, the ventroposteromedial nucleus of the thalamus, because when we get to thalamus, you're going to have, I don't know, 15 to 17 nuclei named, okay? Here's an opportunity to learn one separate from all the others that you're going to be inundated when we cover the thalamus. And that is that these trigeminal pathways, the trigeminal thalamic, the dorsal trigeminal tract, okay, from either the spinal or the principal sensory nucleus, are going to the ventroposteromedial. The somatosensory information is going to go to the ventroposterolateral. So the face and all of the information coming from trigeminal is going to go VPM So if you want to know what portion of the thalamus is dealing with sensory information from the head and neck, okay, the head, really, it's going to be VPM. All right. If you just remember that one, it's one less to remember and will serve as a source of orientation when we start looking at the complex anatomy of the thalamic nuclei. You also have a trigeminal cerebellar pathway and then a trigeminal spinal, again, linking all that facial information to the incoming pain information from the spine. And this diagram, I don't love, but it's all right. And it's a good way of review. You can trace each of the various features coming in and see how they go. It's a reasonably accurate pathway to follow, but it doesn't project well, and I don't want to go over it right. All right, so that's our cranial nerve nuclei, 7, 6, and 5 as we ascend. All right, and now we're going on to look at the rostral pons and the up to the isthmus. So let's start grossly. What do we got? How do you know when you're in the rostral pons as opposed to the caudal pons, for instance? Well, first of all, the fourth ventricle gets smaller as you go up, because in the midbrain, it's going to be the aqueduct of Sylvius. So it's got to get down to that level. So that's the first thing that's happening. The second thing is the cerebellar peduncles are becoming more prominent. The middle cerebellar peduncle and the appearance of the superior cerebellar peduncle. You also, as you move rostrally, you see these transverse fibers of the trapezoid body and the superior olivary nuclei, which are going to be for sound. So we may look at them, but we're not going to talk about them today. You also have the central tegmental tract, which we will talk about in some detail, and the more of the pontine reticular formation, which we will look at. And then you have these other specific rostral pontine nuclei. The locus ceruleus is in the rostral pons. The parabrachial nucleus is in the rostral pons. And you'll see some lateral lemniscus, which is going to be projecting down to the inferior colliculi. And there are some interesting correlations that we'll get to look at here. So this is the appearance of the rostral pons. So get used to that. And the easiest way is the small fourth ventricle. That's sort of what tells you. And then, of course, you have the superior medullary velum. And although we don't want to talk about the trochlear nerve today, you can see the trochlear nerve traveling through the superior medullary velum and exiting from that contralateral to the nucleus. Okay, so let's just work our way around. Here's the MLF. So you can't see. So this is, of course, a Weigert stain. And what you're not seeing are gray matter. All right, so this is going to be our periaqueductal gray. The MLF. Here's the central tegmental tract here. All right. Here's the superior cerebellar peduncle, quite large. All right. Here is the decussation of the superior cerebellar peduncle. We're going to talk about that in a minute. But all of the fibers of the superior cerebellar peduncle are going to decussate in the midbrain or in the very rostral pons. We're all going to do that, okay? Nearly all. The decussate, and so here's the decussation. Here's the, of course, corticospinal, corticobulbar, corticopontine, most importantly. Here are the pontine nuclei. Here are the transverse fibers of the pons as they go from the pontine nuclei to the contralateral cerebellum. All right. Here's the medial lemniscus. Not very medial anymore, is it? And you need to think about this, that what had been a very medial structure in the medulla and in the very rostral pons is now out on the lateral surface. The medial lemniscus is on the lateral surface of the rostral pons. It's way out there, okay? Out here with the spinal thalamic component. All right. Lateral lemniscus, we're not going to be dealing with today. Mesencephalic root of 5. This is the root coming in. We talked about that. Here's the crescent violet, and we see some different things here. We're not exactly at the same level, but very close. So let's go around. Here's the mesencephalic nucleus of 5. All right there. Here is locus ceruleus. All right. a big nucleus. Locus coeruleus is a pretty big nucleus as these things go. Here's the lateral parabrachial nucleus. We're going to talk about that in a minute. And again, this is a big nucleus. These are big, right? Here's the nucleus of the lateral lemniscus, but we're not going to be talking about that today. Here, unstained on a crescent violet, is the superior cerebellar peduncle. Here's the reticular formation, very big and separated by the superior cerebellar peduncle. A lot going on here. And I continue to think of this as useful as a way of thinking about the lower phylogenetic species, which don't have all of the grand demarcated structures that we have, and where their brain is the reticular formation. Okay, I continue to think of that as useful. Pontine nuclei, of course, these are not fibers now, these are the nuclei. Here's the reticulotegmental nucleus, something I don't think we're going to be talking about very much. The superior central nuclei, we're going to talk about those. Here again, in reticular formation, here's the MLF. They put a drawing around it because it ain't, you can't see it, it's not stained. Locus coeruleus. Here's the dorsal nucleus of the raphe. We are definitely going to be talking about the raphe nuclei in a couple of minutes. We don't have time for major detail, but we'll talk a little bit about them. Let's talk about the superior cerebellar peduncle. It carries information from the deep cerebellar nuclei, the dentate, emboliform, and globose. The vestigial is missing because it's not going that way. It's traveling through other pathways to the vestibular nuclei and that sort of thing. It decussates completely in the midbrain. It can synapse either on the red nucleus, or it can make its way all the way up to the ventrolateral nucleus of the thalamus, another nucleus to get used to. So the ventral posterolateral was somatosensory. The ventral posterior medial was the one that we talked about from the trigeminal pathway. The ventral lateral is the one that participates in motor control, all right? It has information coming up through the superior cerebellar peduncle from the dentate especially, but also coming down from the basal ganglia, coming in from the premotor and supplementary motor portions of the cerebral cortex, and those are going to get integrated in ventrolateral nucleus of the thalamus. So we will, again, now we've got 3 nuclei that we should have some sense of before we get to the thalamic lecture. Here again, the superior cerebellar peduncle and the decussation, which is what we want to focus on in this slide. All right. What's in the central tegmental tract? It's named as a location because it's a mixture of stuff. Okay, so it doesn't have a name like a trigeminal thalamic tract because it wouldn't work. It's named by its location because there's a bunch of things going on. First of all, so there are both ascending and descending as well as local fibers. So it really is a mix. The ascending fibers are coming up from the medullary and caudal pontine reticular formation, and they are going to the intralaminar nuclei of the thalamus. Now we're talking about the 4th nucleus that we're dealing with today. The intralaminar nuclei are the activating portion of the thalamus, okay? They are location specific, but not so much modality specific. They are for activating the rest of the brain. So when we talk about, we've been talking about the reticular formation, and I have very carefully avoided the term reticular activating system. But right now, I'm talking about the reticular activating system. So we are going from the medullary and caudal pontine reticular formation to the intralaminar nuclei of the thalamus. This is your reticular activating system. Okay? These are the components of the RAS. Then you have, which are traveling through the central tegmental tract. And then you have descending fibers from the midbrain nuclei going down to the inferior olivary complex. Now that may seem like a very different thing because one's going to cerebellum, but hang on a second, because what were the inferior olivary complex, right? What fibers did they give rise to, right? They gave rise to those ascending fibers that go, the climbing fibers, which enter the cerebellum. What was the purpose of the climbing fibers of the cerebellum? because they didn't carry a lot of information. That's the mossy fibers. What were the climbing fibers doing? They were getting the attention. They were activating the cerebellum. So, whoa. So now we have an ascending pathway for the reticular activating system to the intralaminar nuclei of the thalamus to activate the cerebral cortex, and a descending pathway which is calling the attention of the cerebellum. So actually, although they're very different pathways in some ways, from a functional point of view, they're actually quite similar to one another. That's the central tegmental tract. Okay. And then in addition, you have locally ramifying fibers to integrate the pontine reticular formation. All those scattered nuclei are not acting independently. This is a network, and the network is integrating itself through the central tegmental tract. Okay, so that's what's going on up in that pathway. And here it is. It's not well seen because it's constantly being integrated into the reticular formation. Okay. And there you are. All right, talk about some specific ascending nuclei. This is the parabrachial nucleus. This is there are several. We're going to. I could not find a good picture of the medial parabrachial. Here's the lateral parabrachial nucleus. This is going to carry afferents from nucleus solitarius, but from the caudal portion. What's in the caudal nucleus solitarius? Well, incoming visceral information, especially cardiorespiratory, blood pressure, heart rate, a lot of other visceral information, some metabolic information coming up through cranial nerve 10. So that's all going into the caudal division of nucleus solitarius, which projects to the lateral parabrachial nucleus. Very good. And where does that go? That goes up to the hypothalamus and the amygdala. Well, hypothalamus I get, right? Because I mean, it needs to know all this cardiometabolic stuff, okay? But the amygdala? So when we deal with patients in the ICU, and I don't like this term, so the term that's sometimes used is storming, right? You know, a hypothalamic storm. I prefer a dysautonomia, it's a form of a dysautonomia. But this is probably the pathway that we're participating in. And one of the reasons I think that people like to give bromocriptine, although I think there's not much data to support that. At any rate, then you get to the amygdala. And so to the amygdala is going to be an arousal section for the limbic system and an integrating system. The amygdala needs more attention than previous versions. I'm sure I will be updating the amygdala portion when we get to it later on. The medial parabrachial nucleus brings in the afferents from the rostral nucleus solitarius. And what did that do? Well, that brought in taste. And so it is going, the efferents to the medial parabrachial, from the medial parabrachial are going to go to the thalamus, the hypothalamus, and also to the amygdala. And then there's this interesting, again, I could not find a good picture of the Killica-Fusa nucleus, which is ventral to the parabrachial nucleus, and that is limited to respiration. All right, but so all of this together, medial is for taste, but the lateral parabrachial and the Killica-Fusa bring together all of the cardiovascular information that's coming in through the medulla. And so here's the lateral parabrachial. We previously saw that. It is, again, in its own right, is a large nucleus. This is a cholinergic nucleus primarily. Some of the information coming in, these are going to be coming in through NMDA receptors. There are all sorts of other receptors. Its projections are largely cholinergic. And this diagram, I added several diagrams this morning. Here's a parabrachial nucleus projecting to the amygdala. They should show also one going to the hypothalamus. And the reason that I like this diagram, though, is it shows you the amygdala then synapsing and going to the insular cortex. And we know that insular cortices have a great deal to do with control and sensation of the homeopathic environment of the body. So this is one of the pathways in there, that bringing in all of that information from dorsal motor nucleus, not dorsal motor, but from the sensory nuclei of the vagus. Go on to the locus coeruleus. We've seen some pictures of it already. This is near the periatic reductive gray of the upper fourth. We saw it there. It intermingles with the cells of the mesencephalic nucleus of 5. We saw that. It is a noradrenergic pathway, just exploding everywhere. Telencephalon, diencephalon, other parts of the midbrain, cerebellum, pons, medulla, spinal cord. In other words, the entire central nervous system. It ascends the pathway from the locus coeruleus, ascends lateral to the MLF, and then will go in the medial forebrain bundle. It also ascends in the fornix. This thing goes everywhere. It's got to get everywhere. It will take any route that it can to get where it's going. Its projection to the cerebral cortex, which is mainly to the frontal lobe, is an interesting one because it doesn't go through the thalamus. That's a little peculiar. Not accessing the interlaminar nuclei of the thalamus to do what it's doing. And exactly what it's doing, I think, is not totally clear. We believe that it's involved in modulating sensation, in participating in sleep, and also in activating the cerebral cortex. But how it's doing this and why it's different and why it's not going through the thalamus, I think are things that are still of interest we're going to learn over time. So here's the locus coeruleus, just to focus on that. You can see where it sits. Here's the mesencephalic nucleus again. Here's the periaqueductal gray. So is really sitting close to that. Here it is also, again, sitting adjacent to the periaqueductal gray. The scattered neurons of the mesencephalic nucleus, most of which are going to be rostral to this. I love this stain because it's sky blue. And so you get that it's a nucleus coeruleus, okay? Remember, the locus coeruleus is one of the pigmented structures, and therefore it is involved in Parkinson's disease, all right? And its involvement in Parkinson's disease correlates with depression in Parkinson's disease. And so that has always been part of our understanding of what the locus coeruleus does. When the locus coeruleus degenerates in Parkinson's, seen on anatomy, that patient will have had depression as part of their symptoms of Parkinson's disease. That is, by the way, an intrinsic portion of the disease. All right, and this is a nice diagram again from today. I got these cartoons today. I just was in the mood this morning for adding these cartoons, but I really like this diagram a lot. I think this is a very good diagram and showing you all of the projections of the locus coeruleus. Very excellent. I strongly urge you to take a look at this. Not only does it show you where, but it suggests the kinds of things that are the functions of the locus coeruleus. This is a really good diagram. And then the raphe nuclei. This is a collection of neurons in the median raphe of the pons and medulla. They are part of the reticular formation. They use serotonin. What they all have together is what they all share is that they use serotonin as a neurotransmitter. So you see, we've got these small molecule transmitters, parabrachial using acetylcholine, the locus coeruleus using norepinephrine, raphe using serotonin. What happened to dopamine? We'll get there next talk, okay? Because that's up in the midbrain. So we're not going to be talking about that today. There are other neurotransmitters that are present. There are neuromodulars, but the dorsal nucleus and the nucleus raphe magnus are the serotonergic nuclei, and they're important. The descending pathway from the nucleus raphe magnus is an incredibly important pathway. It is a major inhibitor of pain inflammation coming in. And we will see it when we look at sensation, but these come down and they strongly modulate incoming pain inflammation. And I always think of this when I think about in cancer patients in particular. If you start an opioid for a cancer patient, I really think you should be thinking about adding amitriptyline, because you use amitriptyline to magnify the influence of the nucleus raphe magnus in order to spare the dose of opioid. And you will find many patients who are not controlled on large doses of opioids who with the smallest dose of amitriptyline added in are comfortable on much lower doses. So always think about that. This is the pathway that mediates that extremely important benefit that we should know and use as physicians. And it's involved with the locus coeruleus and sleep. And again, a cartoon on that. This one's been in there. I like this diagram because it shows you all of the various pathways, including the mesencephalic pathway. And you can see how they really extend throughout the neuraxis because they are part of the reticular formation. And just some of the nuclei of the dorsal motor nucleus excuse me, I keep saying that, of the nucleus raffinanus. And a nice diagram again showing you the projections, which in many ways look a lot like the locus coeruleus. I mean, this is quite similar to the locus coeruleus going out to the amygdala and that sort of thing. That's it for today. So we split up the Pons. We made it digestible. We now have 2 lectures. You should spend another hour on this. And in the next talk, we will move up into the mesencephalon, into the midbrain. After we do that, we are going to take a break from new information and have reviews, but reviews with a focus on clinical, okay? And so you, the residents, will be taking over these talks as we will have lectures of strokes in the medulla, stroke in the pons, stroke in the midbrain. And as you do this, please remember that this is a review of neuroanatomy. So you want to absolutely be focusing on the neuroanatomy. Don't worry about the clinical, it will take care of itself, okay? These are clinical correlations, and they'll be very useful and a very enjoyable part of the course that will reinforce all of these raw data that we've been giving you coming in from anatomic studies. All right, so thank you, everybody, and we'll see you in a week.